Good morning, students. Myself, Dr. Raghasudha, Head and Assistant Professor, Department of Microbiology, Government City College, is going to discuss on fermented foods, Part 2. In Part 1, we have discussed about the factors or what are fermented foods and how they are beneficial to our health. In this video, we will be learning about different types of fermented foods. Introduction. Numerous food items are produced by fermentation process in which one or more kinds of microorganisms are responsible for the characteristic flavor, texture and quality of the product. These microorganisms may be the natural foods on the material to be fermented or may be added as started culture. Definition of fermented foods. Fermented foods are those food produced by modification of raw material of either animal or vegetable origin by the activities of the microorganisms. These microorganisms may be of bacteria, yeast and molds, can be used to produce a diverse range of products that differ in flavor, texture and stability from the original raw material. Fermented foods are also defined as those foods which are subjected to action of microorganisms or enzymes to get desirable biochemical changes and cause significant modification to food. If we go through the history and origin of some fermented foods, mushrooms, the approximate year of introduction is 400 BC in China. Soya sauce, 3000 BC in China, Korea and Japan. Wine, 3000 BC in North Africa and Europe. Fermented milk, 3000 BC in Middle East. Cheese, 2000 BC in Middle East. Beer, 2000 BC in North Africa and China. Bread, 1500 BC in Egypt and Europe. Fermented meat, 1500 BC in Middle East. Sourdough bread, 1000 BC Europe. Fish sauce, 1000 BC in Southeast Asia and North Africa. Pickled vegetables, 1000 BC in China and Europe. Tea, 200 BC from China. Worldwide production of some fermented foods. The quantity of cheese produced per annum is 15 million, while as yogurt is 3 million, mushrooms 1.5 million tons, fish sauce 3 lakh tons, dried stockfish is 2,50,000 tons. While beverages, when it is coming to the beverages like beer, it is 1000 million tons, hectoliters, and wine is 350 million HL. Now moving on to the types of fermented foods. Numerous food items are produced by fermentation process in which one or more kinds of microorganisms are responsible for the characteristic flavor, texture and quality of the product. The microorganisms that produce the desirable change may be the natural flora on the material to be fermented or may be added as a started culture. These fermented foods may be classified into, it is only a rough classification of fermented foods, wherein it is classified into bacterial fermented foods and non-bacterial fermented foods. Example for bacterial fermented foods are sauerkraut, dill pickles, sour bread, idli, sausage, green olives, etc. Well, examples for non-bacterial fermented foods are white or common bread and oriental foods. Bacterial fermented foods. Important food items produced in whole or in part by bacterial fermentations are sauerkraut, pickles, sauce, sausage, olives, etc. for human and elisage for animal fodder. Lactic acid bacteria are mainly responsible for the desirable type of fermentation. The type of fermentation in this particular thing is lactic acid fermentation, which is required for the production of each of these substances through other microorganisms, though other microorganisms are also involved in this process. Sauerkraut, this food item results from a fermentation of cabbage by bacteria which are normally present on the cabbage. The cabbage is cleaned, shredded, bruised by tamping and packed with about 2% of salt in jar or barrels. Wooden frames are placed on the cabbage to create pressure. The combined action of salt and pressure draws a considerable proportion of juice from the cells of the leaves. The cells of the leaf respire for a time, rapidly utilize any oxygen present and creates anaerobic conditions. Enterobacter and Erwinia species initiate fermentation, which is accelerated by Leuconostoc mesenteroids producing 0.7 to 1% of lactic acid. Lactobacilli that start multiplying and continue the fermentation to the final stage, increasing the acidity to as much as 2.4% lactic acid. The acids, esters and diacetyls give pleasant aroma and flavor. The fermentation requires 3 to 4 weeks at 21 to 29 degrees centigrade. Similarly, dill pickles are prepared by washing cucumber, packing them in with altering layers of dill leaves, covering with water and adding salt, spices and sometimes even sugar. The soluble sugar of the cucumber gradually diffuses and are fermented with lactic acid bacteria. Early fermentation is initiated by Leuconostoc mesenteroids, Streptococcus faecalis, Pedicococcus cerevisiae, etc., while the final stage is obtained by Lactobacillus brevis and Lactobacillus plantarum. While sour bread is a sourdough from which a starter is saved to involve the next batch, the microorganisms appear to be Escherichia coli and Enterobacter species, which produce a mixed lactic acid fermentation. Idli, which is routinely prepared in our home, is from parboiled rice and decubilated black gram dal, and represents a popular breakfast dish of South India. Its soaken ingredients are thoroughly mixed, little salt is added and the mixture is allowed to ferment overnight for about 10 to 12 hours. Predominant microorganisms that bring fermentation in idli are Leuconostoc mesenteroides, Streptococcus faecalis and Pedicococcus cerevisiae. The fermented material increases in volume and becomes sour. It is then cooked by baking in steam, resulting in soft, spongy product having sour flavor and good taste. Let us see some non-bacterial fermented foods. Example here are white or common bread and oriental food. White or common bread is prepared by moistened flour is mixed with yeast like Saccharomyces cerevisiae and it is allowed to stand for several hours in a warm place. The flour itself contains little free sugar, but there are sufficient quantities of starch splitting enzymes in it to produce some sugar during the leavening process. The sugar is rapidly fermented by the yeast with the production of alcohol and carbon dioxide, the latter causing the rising of the bread. During baking process, alcohol is driven off. While oriental food, in oriental countries like eastern countries of the world, large quantities of diverse foods are prepared from soya bean, wheat and rice through inoculation with members of mucorales, various yeasts and Aspergillus oryzae. Food of the types include temp, a solid food prepared from soya beans processed with species of Rhizopus; Sufu, a Chinese cheese prepared from soya beans and Actinomycor elegans and Mucor species. Miso is also a product of soya beans. It is also known as soya sauce prepared from soya beans and wheat fermented with Aspergillus oryzae and other microorganisms. So let's get an overview of today's video. Today we are going to discuss about some fermented vegetables and fermented milk, and under fermented vegetables we'll go through sauerkraut, olives and pickles, while under fermented milk we'll go through kefir and Bulgarian milk. Coming with Fermented vegetables. Vegetable fermentation originated in the early years of human civilization and even now is widely used by many cultures. Almost all vegetables can be fermented through natural process because they harbor many types of lactic acid bacteria. Worldwide, most of the vegetable fermentation is done domestically. The fermentation process of vegetables can result in nutritious food that may be stored for extended period 1 year or more without refrigeration. Example for some fermented products and vegetables used currently for fermentation are sauerkraut and kimchi from cabbage, olives, cucumbers, carrot, cereals, beans, peas, corns, okra, tomatoes, cauliflowers, peppers, onions, citrons, beets, turnips, radish, chards, Brussels sprouts, and their blends are used are been very good examples for fermented vegetables. Another very good example for fermented vegetable is sauerkraut. The origin of sauerkraut, country origin from Germany. Major ingredients in this are cabbage and salt. Usage, it is used as a salad or side dish. Product description, the fermented shredded cabbage, the product has a sour taste with a clean acid flavor. Microorganisms involved in sauerkraut preparation are Leuconostoc mesenteroides, Lactobacillus brevis, Pediococcus cerevisiae, and Lactobacillus plantarum. The starter culture is a natural microflora. Commercial starter cultures are available. Sometimes black sloping is also being done in preparation of sauerkraut. Let's see the preparation of sauerkraut. It requires fresh cabbage. Then it is the core is taken and trimmed. Then it is trimmed to a thickness of 2 to 5 millimeters. Salt is added. Usually it is of 2 to 3% weight by weight. Then it is filled in wooden vases. After filling in the wooden vases, it is fermented at a temperature of 18 to 20 degrees centigrade for 35 to 40 days. Then it is pasteurized and packed. This is industrial production of a picture of industrial production of sauerkraut, and these are the barrels which are used for the preparation of sauerkraut industrially. Let's see what is happening with the sauerkraut, the fermentation process. The starter for sauerkraut production is usually the normal mixed flour of the cabbage. The raw material has large number of undesirable organisms and a small population of lactic acid bacteria, which is usually less than 1%. Among the lactic acid bacteria, most are Lactococcus species, Leuconostoc species, and small fraction is Lactobacillus species and Pediococcus species. During fermentation, sequential growth of these lactic acid bacteria occurs. The presence of 2.25% salt, large amount of fermentable sugars like sucrose, hexoses, pentoses, and absence of oxygen and low fermentation temperature which is around 20 degrees centigrade facilitates Leuconostoc species, primarily Leuconostoc mesenteroides, to grow rapidly. When the acidity has reached approximately 1%, that is lactic acid, growth of Leuconostoc mesenteroids slows down. Then lactic acid bacteria brevis starts growing rapidly until acid production reaches approximately 1.5%. Then Pediococcus pentososus takes over and increases the acidity to approximately 1.8%. Finally, lactic acid bacteria plantarum starts growing and brings the acid level to approximately 2%. Salting, another ingredient of sauerkraut is salt. The level of salting is critical to obtain a satisfactory product. It must be within the range of 2 to 3% weight by weight, and it is normally about 2.25%. Too little salt, that is less than 2%, and the product softens unacceptable. Too much salt greater than 3% and the correct microbial sequence is not obtained. The salt serves as a number of purposes. It extracts moisture from the shredded cabbage by osmosis to form the brine in which the fermentation will take place. It helps to inhibit some of the natural microflora of the cabbage, such as pseudomonads, which would otherwise cause spoilage, and helps to select for the lactic acid bacteria. It helps maintain the crisp texture of the cabbage by withdrawing water and inhibiting endogenous pectolytic enzymes which cause the product to soften. Finally, salt contributes to the flavor of the product. Let's see the biochemistry of the socket. Leuconostoc species metabolize sucrose, hexoses and some pentoses in the raw material to lactose, acetate, lactate, acetate, ethanol, carbon dioxide and diacetyl. Lactic acid bacteria brevis, it is a obligatory heterofermentative such as Leuconostoc species. It ferments sucroses, hexoses and pentoses to products similar to those by Leuconostoc species. Then Pedococcus pentasauces metabolize hexoses to form mainly lactic acid and some pentoses to lactic acid, acetate and ethanol. Lactic acid bacteria Plantorium also produces products from sucrose, hexoses and pentoses similar to those of Pedococcus pentasauces. The characteristic flavor of socket is a result of the combined effect of the lactate, acetate, ethanol, carbon dioxide and diacetyl in proper amounts. Next, moving on to the another vegetable fermented product is olives. The origin of olives is Mediterranean. The major ingredient of these olives is olive and brine. Usage: it is used as a salad or side dish. The microorganisms involved in producing this fermented olives are L. mesenteroides, Lactobacillus brevis, Pedococcus cerevisiae and Lactobacillus plantarum. The starter culture is usually the natural microflora. Commercial starter cultures are also available. Let us see the preparation of all these fermented olives. Unripe olives are taken. They are light treatment for 10 hours. Unripened fruits are treated with 16 to 2% of lye solution to hydrolyze the glucoside allurobes, then washed and soaked for 10 hours. Then it is fermented in oak barrels with 8% salt, inoculated with Leuconostoc plantarum for 6 to 8 months. The final product has a pH of 3.8 to 4 following up to a 1% of lactic acid production. Let us see the process of fermentation in these olives. The initial pH of the fermentation can be above 7 depending on how much washing was done after the NaOH treatment. As a consequence, the initial microflora during fermentation can include a variety of gram-positive bacilli like Bacillus species and gram-negative enteric bacteria like Enterobacter, Citrobacter, Klebsiella and Escherichia. As organic acids accumulate and the pH decreases below 6, the LAB, principally lactic acid bacteria Plantarum, dominates the fermentation to the exclusion of the other gram-positive and negative microbes. Yeast species may also be present like Candida, Pitchia, Saccharomyces and others and contribute desirable flavor characteristic to the brined olives. Next, moving on to the next vegetably fermented product, those are pickles. The region of origin is North America and Germany. Major ingredients here are cucumber, spices and dill leaves. The usage is salad or side dish. Microorganisms involved are Lactobacillus Brevis, Pedacoccus cerevisiae, Lactobacillus plantarum. The starter culture is natural microflora. Commercial starter cultures are available. Sometimes black sloping is also done for the production of these pickles. Let's see the process. Fresh cucumbers are taken, they are washed, chopped and sliced, then fermented in wooden barrels at 23 to 24 degrees centigrade with 8 to 10% of salt, and they are periodically degassed, gas is removed and packed and finally pasteurized. That's all about vegetable fermented foods. Next, moving on to the next fermented major fermented food is fermented milks. Fermented milk products are also known as cultured dairy foods or cultured dairy products or cultured milk products. These are dairy foods that have been fermented with lactic acid bacteria such as lactobacillus, lactococcus and leuconostoc. The fermentation process increases the shelf life of the product while enhancing the taste and improving the digestibility of the milk. About 2.3% of the total dairy product market is in the form of cultured dairy product. It has great therapeutic benefits and nutritional value also. Let's see a composition of a typical fermented milk as per Codex standards. The constituents are dry matter whose levels are 14 to 18%, protein 2.7%, fat less than 15%, lactose 2 to 3%, lactic acid minimum 0.3% and carbohydrates 5 to 25%. The pH range is about 3.8 to 4.6. Some of the fermented milk products are yogurt, dahi, kefir, kumis, Bulgarian sour milk, acidophilus milk, cultured buttermilk, cultured sour cream, Libin, teat, skyr and cheese. Let's begin with kefir in this particular video. The fermented milk drink that originates in the north causes made with kefir grains, a yeast or bacterial fermentation starter. The organism here, the starter culture is Lactis beta bacterium causicum, which is an yeast, and it consists of, the kefir consists of lactic acid about 0.9 to 1%, alcohol 0.5 to 1%, carbon dioxide 0.03 to 0.07%. There are traces of acetaldehyde, diacetyl and acetone 2 of the total nitrogen. 7% is a form of peptones and 2% of amino acids. Kefir increases vitamin B1, B2 and folic acid in the milk. Manufacture of kefir. Raw milk is taken. It is standardized with a fat of 0.1 to 3.3% in case of Russia, warmed to 65 degrees centigrade, homogenized at 15 MPa, then pasteurized at 95 degrees centigrade for 5 minutes, cooled to 20 to 25 degrees centigrade, inoculated at 20 to 25 degrees centigrade with kefir grains. The kefir grains plus milk are incubated for 20 to 24 hours and ripened for 7 to 8 hours at 10 to 12 degrees centigrade. Then it is strained to remove the ferment without kefir grains. The mother culture inoculated in fresh milk is milk to produce bulk starter culture. Mass fermentation is done for 18 to 24 hours at 20 to 25 degrees centigrade. Finally, it is cooled and stored at 4 degrees centigrade. Next, moving on to the next fermented milk called Bulgarian sour milk. Bulgarian sour milk comes under the general category of yogurt which contains live bacteria. It is a wonderful probiotic food. It is prepared using a combination of two strains, Lactobacillus bulgaricus It is prepared using a combination of two strains, Lactobacillus bulgaricus and Streptococcus thermophilus. Sour milk. Production of Bulgarian sour milk. Sour milk of goat or cow milk is used. It is inoculated with Lactobacillus bulgaricus and Streptococcus thermophilus culture. It is ripened at a temperature of 40 to 45 degrees centigrade, then incubated at 37 degrees centigrade, during which the acidity is increased to 1.4%, and finally it is stored and packaged at 7 degrees centigrade. That's all for today in this video. In the next coming video, we will be discussing about cheese and yogurt production. Thank you.